Hello everyone and welcome to today's session. And today we'll be discussing the NCERT Exemplar questions for the chapter Principle of Inheritance and Variation. This is the part 2 of the lecture. Before starting the lecture, I just want to give you one information that the aim of this section are the materials used in the presentations of education purposes only. We respect the intellectual property rights of the original creators and owners. If you are the owner and have any concerns or issues, please contact us at sathee@itk.ac.in and we shall remove the content immediately. Please note that parts of the content may have been generated using generative AI, which is experimental. So, what are the objectives of today's session? So, we will understand the basic concepts related to the chapter Principle of Inheritance and Variation by discussing NCERT Exemplar questions. Okay, so we'll be discussing in total 28 questions here. So these questions have been divided into these 4 categories: multiple choice questions, very short answer type questions, short answer type questions, and long answer type questions. So let's begin with MCQs first. So what is the first question here? Which of the following will not result in variation among siblings? So in the offerings and in the siblings, there are variations. So they differ from their parents, and siblings differ among themselves also. Clear? So which of the following event will not result in any variation in the siblings? What is the option? Independent assortment of genes. Second option is crossing over. Option number 3 is linkage. 4 is mutation. So what will be the correct answer here? So let's have a look at the answer. The correct answer here is option number C, linkage. So linkage, what is linkage? The genes, when they are present on the same chromosome and when the distance between the genes is very less, then they are inherited together. And because they are being inherited together, they are called as linked genes. And due to this, parental combinations are more. And there is no recombinants which are formed. This recombination leads to variation. So if there is no recombination, there will be no variation. Okay, so in linkage, in linked genes, recombination is very less, recombination frequency is very less. And if the genes are completely linked, then only parental combination will be formed and there will be no recombination, means there will be no variation. Okay, linkage means physical association of the 2 genes and there will be no variation if the genes are linked together. One of the options was independent assortment of the genes. So due to independent assortment of the genes, possible combinations of the different allele is formed, leading to the formation of new phenotype. So this can lead to variation. So this is not the correct answer here. Crossing over involves exchange of the genetic material. This also can lead to variation. So recombination and crossing over, this is one of the main reasons for variations. Mutation, this also is a major cause which leads to variation. So this also causes variation. Next question, Mendel's law of independent assortment holds good for genes situated on. So when Mendel's law of independent assortment is valid, in which case? When the genes they are situated on, option number A is non-homologous chromosome, option number B homologous chromosome, option number C extra nucleogenetic material element, and option number D is same chromosome. What will be the correct answer here? The correct answer here is non-homologous chromosomes. Law of independent assortment holds true when the genes are located on non-homologous chromosomes. Genes located on non-homologous chromosome will assort independently and okay, clear? Genes located on the same homologous chromosome they are not considered to be independently assorted. Okay. Now, if the genes are present on same chromosome, they can be linked, and if they are linked, they are inherited together. So in this case, when the genes are present on same chromosomes, then it will not follow law of independent assortment. Clear? And it will produce only two kinds of gametes because they are linked together. Okay, one of the option was extranuclear genetic material. So extranuclear means outside the nucleus. The chromosome, they are present within the nucleus. Okay, but some of the genes, some of the genetic material is also present outside the nucleus. So these genes which are present, or this material, this genetic material which is present outside the nucleus, they also do not follow law of independent assortment. Clear? So these genes, they have their own inheritance patterns. Okay, they are not subjected to Mendel's law of independent assortment. Now, genes on the same chromosome, they do not assort independently due to linkage. So here you can see, here two cases are shown to you. In the first case, on the left hand side, you can see genes, they are present on different chromosomes. And you can see after meiosis, they are forming 4 gametes. And in the second case on the right, you can see genes are present on the same chromosome. A and B, they are present on the same chromosome. And in the first case, which is on the left hand side, the genes are present on non-homologous chromosome. In the case the genes are present on non-homologous chromosome, A and B, they are present on non-homologous chromosome. Here, genes are independently assorting and 4 types of gametes are being formed. Okay. Clear? These gametes are being formed due to independent assortment. And the image on right hand side, here you can see the genes are linked together and you can see only 2 types of gametes are being formed. Clear? Now, you can see the 2 alleles of a gene A and small a will segregate into gametes independently of the 2 alleles of the other gene, which is B, capital B and small b. So basically what they are saying that the genes, when they are present on non-homologous chromosome, they will segregate. Okay, so in the first case on the left hand side, 4 gametes will be formed, and in the case where the genes are linked, only 2 types of gametes will be formed. The next question, occasionally a single gene may express more than one effect. So you have to name this phenomena. What is this phenomena called when a single gene exhibit more than one effect? Okay, so options are multiple allelism, mosaicism, pleiotropy or polygyny. What will be the correct answer here? So let's have a look at the answer. Answer is option number C, pleiotropy. So usually one gene controls a single trait only, but occasionally a single gene can control or expresses more than one trait. So single gene can affect multiple traits. Like in the case of phenylketonuria, this phenomena is known as pleiotropy. Okay, you can explain pleiotropy with the help of phenylketonuria. Here there is a mutation in one gene. Due to this, an enzyme phenylalanine hydroxylase is not being produced. And because of this, there is accumulation of phenylalanine. And this causes mental retardation, reduction in hair pigmentation, reduction in skin pigmentation. So one gene is affecting multiple traits here. What is multiple allelism? This is not the correct answer here. Multiple allelism. Multiple allelism is actually when a single gene have more than 2 alleles. Okay, when a single gene have 3 or more alleles, you can say that. Example is ABO blood group system. Then what is mosaicism? Occurrence of cells that differ in genetic component from other cells of the body. So usually you can see what is happening. Body is made up of a lot of cells. Sometimes there are some cells in the body and the genetic makeup of these cells differ from the genetic makeup of the other cells. This is known as mosaicism. Okay, this is word is coming from mosaic. Okay. Polygeny means polygenic traits, means the traits which are controlled by many genes. For example, height and skin color. Next question is certain taxon of insects. Some has 17 chromosome and others have 18 chromosome. The 17 and 18 chromosome bearing organisms are. So some of the insects like grasshopper and cockroaches. Okay. In them, the number of chromosomes are different in males and females. So they are saying in some insects, there are 17 chromosomes and they have 18 chromosome in other sex. So this 17 chromosome and 18 chromosome bearing organisms are males and females respectively, female and male respectively. All are males or all are females. What will be the correct answer here? Let's have a look at the answer. So option number A, males and females respectively. So this is an example of XXXO type of sex determination system which is seen in grasshoppers and cockroaches. Here what is happening, the males have only one X chromosome and the females have a pair of X chromosome. Okay, so females have one chromosome more as compared to males. Clear? So females will be having 18 chromosome here and males will be having 17 chromosomes. Clear? So XO means there is an absence of one sex chromosome. So males lack one X chromosome. They have only one X chromosome. So they have in total 17 chromosomes and females have in total 18 chromosomes because they have a pair of X chromosome. The next one, the inheritance pattern of a gene over generations among humans is studied by the pedigree analysis. Clear? Now, characters studied in pedigree analysis equivalent to. What are the options? Option number A is quantitative trait, B is Mendelian trait, C is polygenic trait, and D is maternal trait. What is the correct answer here? The correct answer is option number B, Mendelian trait. So pedigree analysis used to study the inheritance pattern of traits that follow Mendelian inheritance pattern. The genes, they are inherited according to Mendel's law of inheritance. Clear? Mendelian traits are controlled by single gene, okay, with clear dominant and recessive alleles. Clear? So this, the characters that you study in the pedigree analysis is equivalent to Mendelian traits, which are controlled by single gene. One of the options was quantitative traits. So quantitative traits, they are polygenic traits. They are controlled by more than two, you can say one gene. So, or they are controlled by multiple genes. And they have additive effects. Clear? Polygenic traits, again, this is also, you can say, controlled by many genes. So they are like quantitative traits only. Clear? They are exceptions to Mendel's rule. Polygenic traits also known as quantitative traits. Maternal traits, they are traits inherited and expressed from the maternal parent to the subsequent offerings. So when the mother is transmitting the, okay, or transferring the traits to the offerings, it is known as maternal traits. The next question, it is said that Mendel proposed that the factor controlling any character is discrete and independent. This proposition was based on what are the option? Option results of the F3 generation of the cross. Option B, observations that the offerings of a cross made between the plants having two contrasting characters shows only one character without any blending. Option number C, self-pollination of F1 offerings, and option number D, cross-pollination of F1 generation with a recessive parent. So what would be the correct answer here? So let's have a look at the answer. So here the answer is option number B. Okay, observation that the offerings of a cross made between the plants having two contrasting character. Two contrasting character means they are talking about a dihybrid cross. Okay, the offerings show only one character without any blending. So there is no blending of the traits. Okay, clear? So there is no blending of the character. So when Mendel did experiment, he didn't observe any intermediate phenotype. Okay, clear? So the characters that Mendel chose, that had contrasting traits. They were controlled by only single gene, and one gene has two alleles. So you can see here, this is a dihybrid cross. Okay, this is a cross between a plant having round and yellow seeds with plant having wrinkled and green seeds. The genotype is also written. Clear? Then the gametes produced by each of the parent is written here. One of the parents is homozygous dominant, other parent is homozygous recessive. Then the F1 generation is heterozygous, having capital R small r, capital Y small y. You can self the F1 generation to get the F2 generations. In the F2 generation, each F1 parent will produce 4 kind of gametes. And if you analyze the phenotypic ratio in the F2, it will be 9:3:3:1, and genotypic ratio will be 1:2:2:4:1:2:1:2:1. So there are 9 different genotypes which are formed and 4 different phenotypes which are formed. Okay, so here the genes are independently assorting. Clear? That's why you're getting new combination in the F2 generation. Okay, they are following law of dominance also. In the F1 generation, you get only the dominant phenotype in the F1 generation. Okay, and no intermediate phenotype is being observed here. Okay, so you can see results of a dihybrid cross were 2 parents differed in 2 pair of contrasting traits, seed color and the seed shape. So after dihybrid cross, you can see you get the parental combinations and you get the recombinants as well. Clear? The rest of the options are false. Now, what is given to you? 2 genes A and B are linked together in a dihybrid cross involving these 2 genes. Okay, A and B genes. A and B are linked together. Now, the F1 heterozygote is crossed with the homozygous recessive parental type. So, this is a kind of test cross. The F1 heterozygote, when it is crossed with homozygous recessive parent, this is test cross. Okay, this is a dihybrid test cross because 2 genes are involved here. So, what would be the ratio of offerings in the next generation? These are the options given to A, B, C, D. What will be the correct answer here? So this is a dihybrid cross. You can see that you can eliminate option number C and D because these are the ratio that you get in monohybrid cross. Okay, and another you can see this is a kind of test cross. So what will be the correct answer here? Clear? So here you can see you are getting option number D here. Okay, so let's see how you got this 1:1 ratio. So F1 heterozygote genotype is, so this is the genotype of the heterozygote. Capital A, capital B, small b. Homozygous recessive is small a, small b, small b. These are the genotype of the parents, you can say, actually. So since genes are linked, gametes found by F1 will be, okay, so these are the gametes formed by the F1 generation. AB, it will form the parental type, and small a, small b, these are the parental type. So actually they are saying that the two genes are linked together. Okay, these are linked together. That's why you are not getting 1:1:1:1 ratio. Otherwise, you would have got 1:1:1:1 ratio. But the genes are linked together, okay? That's why you are getting 1:1, option number D. Okay, otherwise you have got the ratio of 1:1:1:1 in a dihybrid cross. Okay, clear? So as they are linked, when the genes are linked together, then they are inherited together. Only two kinds of gametes will be formed by the F1 generation. So these are both parental types because there is no crossing over. So you can make a Punnett square and you can see you are getting 1:1 ratio. Okay, you are getting the parental type only. After crossing, you are getting parental type only. Clear? So this is 1:1 ratio or 50% ratio. Now next, in the F2 generation of a Mendelian dihybrid cross, the number of phenotypes and genotypes are. Again, they are saying this is a Mendelian dihybrid cross. And here the genes are not linked, they are independently assorting. So what will be the phenotype and genotype in the F2 generation? So these are the four options given to you. You have to choose the correct answer from these options. Okay, so 4 phenotypes and 9 genotypes. This is the correct answer. So again, you can see you can make a dihybrid cross here. Okay, you can make a cross between a plant having round and yellow seeds with a plant having wrinkled and green seeds. You can write the genotype. One parent is homozygous dominant and the parent is homozygous recessive. Then you can write the gametes which are found by each parent. Then the F1 will be heterozygous. Clear? Selfing of the F1 is done. You can write the gametes of the parent, gametes of male on one side, gamete of the female on other sides. 4 types of gametes will be formed here. Okay, you can calculate the number of gametes using the formula 2 raised to power n, where n is the number of the heterozygous pair. So in the F1, you have 2 heterozygous pair. The total number of gametes produced will be 4. Now, after analyzing this, you can say Punnett square in the F2 generation, you can see you are getting 4 phenotypes in the ratio of 9:3:3:1, and you are getting 9 genotypes. Here the phenotypic ratio, genotypic ratio is also mentioned. So you should remember both phenotypic and genotypic ratio in the F2 generation. Clear? What are the phenotypes that are formed? Round yellow, round green, wrinkled yellow, and wrinkled green. And 9 genotypes are formed. Now another question. So here they are saying mother and father of a person with blood group O. Clear? Have A and B blood groups respectively. What would be the genotype of both mother and the father? You have to calculate the genotype of both mother and the father. Okay, so these are the 4 options given to you. Option number, mother is homozygous for A blood group and father is heterozygous for B. Okay, homozygous for A means the mothers will be having IA and IA. Okay, father is heterozygous means father will be IB and small I. Then option B, mother is heterozygous for A and father is homozygous for B. Option number C, both mother and father are heterozygous for A and B blood group respectively. Option number D, both mother and father are homozygous for A and B blood group respectively. So what will be the correct answer here? So the person is having O. What will be the genotype of mother and father? So let's have a look at the answer. So you can see both mother and father will be heterozygous for blood group A and B. So why this is so? Because the child is having O blood group. O blood group means it will be having small I alleles. So this person with blood group O, this person will be homozygous recessive for small I. So this means both mother and father will have at least one I allele. That's why the child is inheriting one I from father, one I from the mother. So mother and father should be heterozygous for A and B. Okay. So the father should be having IA small I, mother should be having the genotype IB and small I. Clear? Now very short answer type questions. How are alleles of particular gene differ from each other? You have to explain its significance. So let's have a look at the answer. So alleles are just alternate form of the gene. They are polymorphs. The two alleles, they differ only in the nucleotide sequence. A gene have two alleles. Suppose there is a gene for height. There are two alleles for that, capital T and small t. These two alleles differ from each other in just nucleotide sequence. So there is a very minor difference in the nucleotide sequence of these two alleles. Okay. So this result in contrasting phenotype. So one becomes dominant, one becomes recessive. One is controlling tallness, one is for dwarfness. And you can see alleles are the alternative form of gene, same gene. Clear? So height, if you consider height as a trait, it has two alleles, capital T and small t. Okay. Now, what is the significance? So significance is genetic variation. Okay. So it leads to genetic variation. Alleles contribute to the genetic diversity within a population, and it means that individuals within a species can have different combinations of the allele for the same gene. So a diploid organism can inherit two alleles. So it can inherit different combinations of the allele. So the person can inherit capital T, capital T, capital T, small t, or small t, small t. So this is, you can see, this is leading to variations. So there are a lot of phenotypic variations. Clear? So in case of small t, the plant will be dwarf. In case of capital T, the plant will be tall. In case of capital T, small t, again the plant will be tall. But in the case of incomplete dominance, the heterozygous capital T, small t can be of intermediate height as well. Okay, clear? Example is eye color or blood type. Okay, blood type is a very good example. So there are three alleles for the gene I. Gene I controls the blood group system. So there are three alleles, so different combination of these three allele give rise to four phenotypes, which is blood group A, blood group B, blood group AB, and blood group O, and give rise to six genotypes. The next question, in a monohybrid cross of plant with red and white flower plants, Mendel got only red flower plants. Okay. On self-pollinating this F1 plant, Mendel got both red and white flower plants in 3:1 ratio. You have to explain the basis of using capital R, and small r symbols to represent the genotype of plants of the parental generation. So why you are using capital R, and small r to represent the genotype? You have to explain this. So you can see here, capital R is for red color, small r is for white color. Okay, so this is basically done to avoid confusion. So instead of writing W for white, you are writing capital R. This is being done to show that these two alleles, they belong to a single gene, capital R and small r. If you write capital R for red and W for white, then you might confuse this with two different genes. Okay, that's why you write capital R and small r. So one parent is homozygous dominant, another parent is homozygous recessive. Then capital R parent, this will produce gametes having capital R alleles. Small r parent will produce gametes that will be having only small r allele in them. Then the F1 generation will have the genotype capital R, small r. You can after selfing of the F1 generation, what you will get the 3:1 ratio. You will get the 3:1 phenotypic ratio here. So 3 will be red here, which is the dominant trait, and 1 will be a recessive trait, which is white here. Okay, recessive trait is only observed when in the homozygous condition, and both the recessive alleles are present together. So here you get the phenotypic ratio 3:1 and genotypic ratio is 1:2:1. Now, generally, uppercase letters are used as symbols for dominant and lowercase symbols are used for the recessive trait. Clear? And in the F2 generation, you get 3:1 phenotypic ratio. Now, both these parents are true breeding lines. Okay, true breeding line, they are homozygous for the particular traits. Clear? So they are homozygous, and how they are represented? One of the parent is represented as capital R, capital R. Okay, so this will be having the red color flowers, and the other parent will be smaller. Okay, which will have white flowers. So both of them are homozygous. Both of them are true breeding lines. Okay, clear? Now, next one, for the expression of traits, genes provide only a potentially, and the environment provides the opportunity. You have to comment on the versatility of the statement. So what is the correct answer? Obviously, genes are not the only factors that determine the phenotype. Okay. What else determine the phenotype? Environment also play a role in the expression of any trait. Genes are actually quite active throughout our life, switching their expression on and off in response to the environment. So some of the genes are also affected by the environment. The polygenic traits, they are also affected by the environment. The phenotype, you can say, is also affected by the environment. Okay. So phenotype, you can see the final phenotype is the combination of the genotype, which is the potentiality, and the environment, which is the opportunity. Beside the effect of internal factors like hormones and metabolism on gene expression, external factors like temperature, like nutrition, etc., also affect the gene expression and ultimately exhibiting the phenotypic changes. For example, skin color. Skin color is controlled by multiple genes, and skin color is also affected by the environment. So if the persons go in sunlight, then the amount of melanin increases and the color of that person becomes dark. So we can say that genes provide only the potentiality and the environment provides the opportunity for the expression of the trait. Now next, ABD, these are three independent assorting genes with their recessive alleles, small a, small b, and small d. Respectively, a cross was made between individuals having this genotype, capital A, small b, capital D, with an individual having this genotype, small a, small b, small d, small d. So these are homozygous recessive for three alleles. You have to find out the type of genotypes of the offerings which are produced. So what will be the correct answer here? Let's have a look at the answer. So the given cross between these two individuals, this is a tri-hybrid cross because 3 genes are being involved here. Accordingly, the type of offerings produced would be, so you can make a cross here. This is the cross showing you a tri-hybrid cross. So you can calculate the gamete with the formula 2 raised to power n, where n is the number of heterozygous pairs. So in the first parent having the genotype capital A, small b, capital D, there is only one heterozygous pair, so it will produce only 2 gametes. And the other parent, which is homozygous recessive for all the genes, 3 genes. Here there is no heterozygous pair. All are homozygous recessive. So you can use the formula 2 raised to power n here. Here L will be 1 here. So it will be producing only one type of gametes. Now you can, okay, after fertilization, you can see 2 types of offerings are being formed here. Okay. So you can see the gametes, 2 gametes produced by the parent. Okay, 2 gametes are what? Capital A, small d, another is small a, small b, small d. So they have equal chance to fertilize the other gamete, which is produced by the other parent. 2 types of offerings are being produced. In the society, woman is often blamed for bearing male child. Okay, or for not bearing male child and for bearing the female child. Do you think it is right? You have to justify the statement. Okay. So sex is controlled by males. The gender of the unborn child is determined by the male, not by the females. So this is very unfortunate that the females are blamed for giving birth to the female child, but it's actually the males who determine the gender of the child. So males are heterogametic. Always the heterogametic sex determines the sex of the children, sex of the offerings. So in case of humans, in humans we have XX and XY sex determination system. Males are heterogametic. They produce 2 kinds of gametes because males have XY in them. So half the sperm have X chromosome, half the sperm have Y chromosome. Females are homogametic. They produce only 1 kind of gametes. Okay, because females are X and X. All the eggs that are produced by the females, that will consist of only X chromosome. Okay. Clear? So now you can see males are producing 2 kinds of gametes. So if the sperm having X chromosome, this fuses with the egg, then the zygote, in the zygote there will be XX condition. This will develop into female child. And if the sperm having Y chromosome, this fuses with the egg, then the zygote will have XY genotype and this will develop into the male child. Okay. So you can see this is a sex determination system in the humans. Males are XX and females are XY. Males are producing 2 kinds of gametes. Females is producing only 1 kind of gametes. And there is an equal chance that the female can give birth to male child or female child. Okay, so this is a genetic makeup of sperm that determines the sex of the child. Yeah, and it is also evident that in each pregnancy, there is always 50% probability of having either male or female. Next question, you have to discuss the genetic basis of wrinkled phenotype of the pea seed. So what is the answer here? The genetic basis of wrinkled phenotype of pea seed is as follows. So you know seed shape is determined by a single gene. Single genes controls the seed shape. Now, where the allele capital R is for round peas and it is dominant over small r, and small r is for wrinkle seed, which is a recessive trait. If the allele of the genes controlling the seed shape are homozygous, then what will happen? It will show the character or phenotype of the same alleles. So, for example, capital R, this is homozygous condition. These seeds will be round, and smaller, this is also homozygous condition. These will be wrinkle seeds. Then in the case of heterozygous, what happens? The dominant phenotype is observed. In the case of capital R, small r, this will be round seeds because capital R is dominant over small r. Okay, next question. Even if a character shows multiple allelism, an individual will only have two alleles for that character. By so see, a diploid organism have only a pair of alleles. One allele it inherits from the father, another allele it inherits from the mother. So although sometime in some cases a single gene can have three alleles, like in ABO blood group system, a single gene can have multiple alleles. But still the organism, the diploid organism will have only two alleles. So why is this so? So multiple alleles are the multiple form of gene which occur on the same gene locus. Locus is the position of that gene, location of the gene on the chromosome. An individual will have only two alleles in case of multiple allelism because, so first of all, individuals develop from the zygote, and zygote develops due to fusion of the sperm and the egg. Now, sperms and egg, they are haploid and they consist of only one allele. Sperm and egg, they consist of only one allele. And when they fuse together, they form the zygote. So zygote will have only two alleles, one from sperm, one from the egg. Okay. Now, next question, how does a mutagen induce mutation? You have to explain with the example. So mutagen, what are mutation? Mutagen induces mutations. And how they induce mutation? By changing the base sequence or by insertion or deletion or substitutions. Clear? So mutations, how they induce this mutagen, how they induce mutations? By causing insertion of a nitrogenous base, deletion of a nitrogenous base, or substitution of a nitrogenous base. Examples of mutagens are, mutagens may be physical, for example, ionizing radiation, X-rays, UV rays, gamma rays. They can be DNA reactive chemicals. Okay, for example, hydroxyl radical, peroxide, H2O2. H2O2 is hydrogen peroxide. Okay, so these molecules can also cause mutations. They can react to DNA and cause mutations. Now, there are some viruses also which can cause mutations. Okay, so there are three mutagens: physical mutagens, chemical mutagens, and biological mutagens. Okay, sickle cell anemia is an example of point mutation. So what happens in sickle cell anemia? There is a single base pair substitution at the 6th position of the beta globin chain. And due to this, there is an amino acid substitution. Glutamic acid is replaced by valine. Okay. Clear? So you can see GAG changes to GUG. GAG codes for glutamic acid, GUG codes for valine. So this change, this amino acid substitution occurs at the 6th position of the beta globin chain. Okay. So due to this, what happens? When the glutamic acid is replaced by valine, the hemoglobin polymerization takes place and the RBCs become sickle shaped. Clear? So usually the RBCs are biconcave in shape, they become sickle shaped. Next question, why is the frequency of red-green color blindness in many times higher in males than in the females? So why is color blindness common in males as compared to females? What will be the answer here? So you can see color blindness, it is X-linked sex inheritance. Okay, it is X-linked recessive inheritance actually. So this mutation occurs on the X chromosome. And males, they have XY chromosome, and females have XX chromosome. So females are usually carriers. Clear? So as the females have X and X chromosome, so what happens? Even if a mutated gene is present on the X chromosome, so there is a color blindness gene present on the X chromosome, but there is a normal gene present on the other X chromosome. That's why females are usually carriers, and they are unaffected. Female can only be colorblind if she inherits both the X chromosome from the parents, and both of the X chromosome should carry the color blindness gene, which is quite rare. Males are XY, so if they inherit one X chromosome having the color blindness gene, that is enough to exhibit the disease, to exhibit the color blindness, because they don't have a normal, you can say, gene on the Y chromosome. So males are usually affected as compared to females. All right. So this we have discussed. Now the female must have the allele for it in both her X chromosome, then only she will be affected. When only one X chromosome of female possesses allele for a color blindness, she is said to be carrier. But males develop color blindness even when their sole X chromosome, so they have only one X chromosome. So even if one X chromosome carries the color blindness gene, the males will be colorblind. Okay, thus males are more prone to color blindness, while the females are the usually carriers. Next question, if a father and son are both defective in red-green color vision, okay, father and son, both of them, is it likely the son inherited the trait from his father? You have to comment. Okay, so you know color blindness, we know that it is X-linked recessive condition. It is X-linked recessive disorder. Now you can see father will only transmit Y chromosome to the sons, not the X chromosome. So sons, the males, they receive their X chromosome from the mother, not from the father. So if the father is having color blindness, it will not transmit color blindness to the son, because it is only transferring the Y chromosome to the son. The color blindness gene should be present in the mother, should be inherited from the mother, then only the son will be colorblind. So mother should be at least the carrier. Yeah, so male to male inheritance is not possible for X-linked traits, because male only transfer Y chromosome to the sons. Yeah, so if the male is colorblind, if the father is colorblind, then it cannot transfer colorblindness to the males. So in the given case, the mother of the son must be carrier for colorblindness gene, and the father is colorblind. Clear? Now for this, mother is transmitting the gene to her son. Okay, the father will only transmit Y chromosome to the son, and if the son is affected, he has inherited X chromosome having colorblindness genes from the mother, not from the father. Now discuss why Drosophila has been extensively used in genetical studies. This is the next question. Now Morgan was working on fruit flies whose scientific name is Drosophila melanogaster. So Drosophila melanogaster is the model organism. It is found suitable for genetic studies due to the following characteristic. First one, they can be grown easily on simple synthetic medium in the laboratory. They complete their life cycle in about 2 weeks, so we can analyze the progeny also. Okay, it is producing progeny in just 2 weeks, so we can study the inheritance of the traits from parents to the offerings in just 2 weeks. A single mating could produce a large number of progeny flies. Okay. So, clear? Here, the single meeting you can see producing large number of progeny flies, or large number of offerings are being produced. So you can study a lot of variations in this large number of progeny. Then there is a clear differentiation of sexes in Drosophila. Male and female flies are easily distinguishable. It has many type of variations that can be seen with low power microscope. So Drosophila exhibit lot of variations, and these variations can be easily seen. This can be easily observed under the microscopes. So you can study these variations under the microscope. You can see this is the image showing you Drosophila melanogaster. Image A is that of a male, so males are smaller as compared to females. Next question, how do genes and chromosomes share similarity from the point of view of genetic studies? So what is the similarity between genes and chromosome from the point of view of genetical studies? Okay, so by 1902, chromosome movement during meiosis has been worked out. And Water and Sutton, Sutton and Bovray, 1902, noticed that behavior of chromosome was parallel to the behavior of genes. Means both of the chromosome and gene, they behave very similarly. They behave in similar manner. Their behavior is parallel to each other. Okay, if the chromosomes are segregating, in a similar way, genes will also segregate. Chromosomes are present in pairs, genes are also present in pairs. Chromosomes segregate during cell division, genes are also segregating during the cell division. Why this is happening? Because genes are present on the chromosomes. Okay. So Sutton and Bovray, they gave chromosomal theory of inheritance, and they use chromosomal movement to explain the Mendel's law. So how they explain Mendel's law of inheritance? By chromosome movement. Clear? Now, they study the behavior of chromosome during mitosis, which is known as equational division, during meiosis as well. Yes, so they were studying the chromosome segregation, chromosome movement during the cell division. And they said chromosomes are a physical structure which can be used to explain the inheritance, Mendelian inheritance. Now, the chromosome as well as genes occur in pairs, and the two alleles of gene pair are located on the homologous sides of the homologous chromosomes. All right. So you can see this is an image showing you chromosome movement during the meiosis. In the G1 phase, you can see these are the chromosomes. Now, in the S phase, there's a DNA duplicates. In the G2, you can see DNA have duplicated, chromosomes have duplicated, DNA content has duplicated. In the anaphase 1 of meiosis, chromosomes separate. In the anaphase 2 of meiosis, chromatids are separating. In the end, 4 cells are formed. So genes are segregating, and similarly, chromosomes are segregating, and similarly, genes also segregate because genes are present on the chromosomes. So Sutton and Bovray, they explained Mendel's law of inheritance by chromosome movement. Clear? So you can see in the end, 4 cells are formed at the end of the meiosis. And the chromosomes segregate when the gametes are formed. Similarly, the alleles also segregate independently during gamete formation. Next question is short answer type question. What is recombination? You have to discuss the application of recombination from the point of view of genetic engineering. So recombination refers to the generation of new combination of genes. So in recombination, what happens? There is a exchange of the genetic material. Due to this exchange of genetic material, because of the exchange of genetic material, new combinations of the genes are formed. Okay, so recombinants are found which is different from the parents. So these leads to variations as well. Clear? The crossing over occurs during the Pachytene stage of meiosis. and meiosis, you can say, takes place during the gamete formation. What are the application of recombination? So through recombination, you can say new combinations are introduced, means variations are introduced. These variation can give rise to new traits, and this variation can accumulate over several generations and lead to evolution of a new species as well. So this recombination increases variability, which is useful for natural selection and under change environment. So some of the, you can say, variations that are produced through recombination, some of these variation can give rise to new traits, and these traits can help in the survival of these species when the environment changes. So this helps in the natural selection as well. So only those organisms will be selected that consist of these variation, because they are giving survival advantage to that species. Frequency of crossing over depends on the distance between the two genes. Okay. So the percentage recombination or frequency of recombination is more if the genes are, or if the distance between the gene is more. And this is, you can say, percentage recombination is also used to calculate the distance between the two genes. And this can be used for making genetic maps. Clear? Now it has been proved that genes, they lie in a linear fashion on the chromosomes. Clear? So during crossing over, there is the exchange of the genetic material, or you can say there is the exchange of the genes. So due to this, new combinations of the genes are formed. Okay. Clear? Now breeder have to select small or large population for obtaining the required crossovers. And for obtaining crossover between closely linked genes, a very large population is required. So you can see with recombination, new combinations are being produced. And when the genes are linked, then recombinants are very less. Okay. If you want to get recombinants when the genes are very closely linked, then you need a very large population for that. Here, now useful recombinants are selected by the breeders to produce new varieties. Okay, so deliberately, you can see the breeders, what they do. These breeders deliberately select the variations that give survival advantage, or these variations can give rise to new traits. So they breed, these breeders, they select these, you can say, advantageous variations, because this variation can give rise to new traits as well. So they can pick those varieties which have these variations, which have arisen due to recombination. Now, what is artificial selection? Do you think it affects the process of natural selection? How? Okay, so artificial selection is also known as selective breeding. So this describes intentional breeding for certain traits or combination of traits by humans for exploiting the various existing among variations existing among the species. So in artificial selection, what is done? Parents are selected which are having desirable characteristic, and then these parents are crossed with each other. Okay, in a hope that the hybrid which will be formed, that will be having the characteristic which were present in the parents. That's why selection of the parent with desirable character is very important here. Okay, so when you cross, you expect that the F1 generation or the hybrid which is being produced will have the traits which are present in the parents. Okay, this is known as selective breeding or artificial selection. There are three types of artificial selection: mass selection, pure line selection, and clonal selection. We will not go in detail. Okay, and this artificial selection affects the process of natural selection. Okay, natural selection is slow, artificial selection is fast, because you are intentionally, you are breeding, intentionally you are crossing the parents. Natural selection selects for or even against the traits based on their effect on fitness of the organism. Okay. But in artificial selection, you want the traits that are beneficial. Okay. Natural selection can select against also the trait, for or against the traits. Okay. Artificial selection traits are selected based on human preferences for improving the trait. Clear? So in artificial selection, parents are selected in such a way that the hybrid that you get can be used for human benefits as well. Process of natural selection leads to evolutionary change in the expression of the trait in the population, whereas artificial selection, though being the same process, involves the trait preferred by humans for its own benefit. So artificial selection is done by humans because we want to create varieties that can benefit humans. But in natural selection, okay, it leads to evolution, but that is very slow. And that is not, you can say, this natural selection is not based on human preferences. Artificial selection is based on human preferences. Okay. It is much faster. Artificial selection is much faster than the natural selection. Now, with the help of example, differentiate between incomplete dominance and codominance. So, incomplete dominance, what happens in incomplete dominance? It is a phenomena where two contrasting alleles are present together. In incomplete dominance, the heterozygous have intermediate phenotype, which is different from both the parents. Okay. Classical example of incomplete dominance seen in the flower color of snapdragon. Here in the F1 generation, pink flowers are produced. Okay, in the heterozygous condition, pink flowers are produced, although the parents were having red color flowers and white color flowers. Okay, what is codominance? Where both the alleles are expressed in the heterozygous condition. Example is ABO blood grouping system. Okay, ABO blood grouping system, where allele IA and IB both are codominant. Clear? So you can see you are getting the AB blood group. In AB blood group, both the alleles are present, IA and IB. Now, next question, it is said that the harmful alleles get eliminated from population over a period of time, yet sickle cell anemia is persistent in human population. Why? Okay, so sickle cell anemia, it is autosomal recessive disease. Here, this is caused by when the hemoglobin polymerizes and the RBCs become sickle-shaped and they cannot carry the oxygen. So it can be lethal, but the sickle cell shape of the RBCs protects from the malaria because malarial parasite cannot, you can say, replicate in the sickle-celled RBCs. This allele is most common in people of African descent. Okay, and in that region, malaria is quite prevalent. So the malarial parasite, malarial pathogen cannot grow, cannot replicate in the sickle cell shaped RBCs. Clear? So they don't get malaria. So it provides protection from the malaria. So individuals with HbA, HbS heterozygotes tend to survive better than individuals with HbS, which are homozygotes. Clear? So the person having heterozygous genotype, HbA, HbS, who are the carriers, they will survive. And the individual which are homozygous for the HbS allele, which means the person that will be having HbS, there are chances they will not survive. So that's why sickle cell anemia is still persisting in the human population, because it is providing, you can say, protection from the malaria. Clear? Now, a normal visioned woman whose father is colorblind marries a normal visioned man. What would be the probability her sons and daughter to be colorblind? You to explain using a pedigree chart. So you can see this is a pedigree chart that is shown to you. Female will be a carrier here. because her father was colorblind. Male is normal. So you can see, you will get all of these offerings. So male will only transmit Y chromosomes to the sons, but it can transmit X chromosome to the female, to the daughters. The mother can transmit the X chromosome having color blindness genes to male as well, to male also, and to the female as well. Okay, so we will get all these combinations in the offerings. Okay, so according to the pedigree chart, one of the two daughter is carrier, but both are normal vision. Probability of a daughter to be color blindness is 0%. Okay, among the son, one of two is colorblind, and probability of son to be colorblind is 50%. Okay. Now discuss in detail contribution of Morgan and Sturtevant in the area of genetics. So Morgan, he gave linkage and recombination. Okay, Morgan, he was given Nobel Prize in 1933. Okay, Morgan was carrying out dihybrid crosses in the Drosophila melanogaster, and he gave the chromosomal theory of linkage. So he said the genes, when they are present on the same chromosomes and when the distance between the two genes are less, they are linked together, they are inherited together, and parental combinations will be more. Okay, he also described recombinations, which is what? Which is the exchange of the genetic material. So he established the principle of linkage, crossing over, sex-linked inheritance, and discovered relationship between gene and the chromosome as well. Okay. He also worked on the mutation also. Okay, let's talk about Alfred Henry Sturtevant. He was a student of Morgan. Okay, he was given the National Medical of Science in 1960. He was given the National Medical of Science in 1967. What was his contribution? So he created the first genetic map of a chromosome while working on Drosophila genome. So what he did, he used the recombination frequency to calculate the distance between the genes. Okay, so his main contribution to science included analysis of the genetic linkage groups, which became classical method of chromosome mapping that is still used today. Clear? So basically he used recombination frequency to calculate the distance between the genes, and he used this to create the genetic maps. In 1913, he determined that genes were arranged on the chromosome in linear fashion, like beads on a necklace. He also showed that the genes for any specific trait was in a fixed locus. Means genes are present on a fixed position on the chromosome. Locus means position. His work on Drosophila proved that two closely related species showed newly recurring mutations that were allelic and thus probably identical. Okay. His work also helped to determine genetic role in sexual selection and development, and displayed the importance of chromosomal crossing over in the mutations. One of the Sturtevant principal contribution was the introduction to the concept that the frequency of crossing over between two genes could help to determine their proximity on a linear genetic map. So he used recombination frequency to calculate the distance between the genes. His experiment determined that the frequency of double crossing over can be used to reduce the gene orders. So this was okay. Some additional work done by Sturtevant as well. Now you have to define aneuploidy. How is it different from polyploidy? You have to describe the individuals having following: trisomy, XXY, and XO. Okay, trisomy of 21 chromosome is basically Down syndrome. It is aneuploidy. It is caused by non-dissection of the chromosome. Okay, this is 2n plus 1 conditions. So the person will have one chromosome, one extra chromosome here. So total number of chromosome will be 47 in case of Down syndrome. Okay, clear? It is different from polyploidy. In polyploidy, there is increase in entire set of chromosomes. Then XXY condition. This is a chromosomal abnormality. Down syndrome is also chromosomal abnormality. It is a trisomy of the 21st chromosome. Means there are 3 pair of 21st chromosome. Characteristic of Down syndrome. Okay, short stature with round head, open mouth, protruding tongue, short neck, slanting eyes, broad, short hands. So these are some symptoms of Down syndrome. Person will be mentally retarded. Physical growth will be, you can say, retarded, underdeveloped gonads and genitals. These are the image showing you child having Down syndrome. Okay, then Klinefelter syndrome. Here the karyotype is 44 plus XXY. So again, there are 47 chromosome here. There is an additional X chromosome in the males. Characteristic of Klinefelter syndrome, overall masculine development, but the male possesses feminine character like development of breast, which is also known as gynecomastia. Okay, poor beard growth, feminine pitched voice, the person will be sterile. Okay, Turner syndrome is X0 condition. This occurs in females who lack one X chromosome. So karyotype will be 44 plus X0. What are the characteristics of Turner syndrome? Okay, the female will be sterile with rudimentary ovaries. Okay, shield-shaped thorax, webbed neck. Poor development of breast, short stature, small uterus, puffy fingers. So this is an image showing you person having Klinefelter syndrome and Turner syndrome. Okay, so this is the end of the lecture. I hope this lecture was quite knowledgeable for you. We'll meet you in the next session. Thank you very much for listening.